I'd like to introduce our panel. To the far left is Dr. Rick Rudick, who is himself a longtime MS researcher. Rick is the director of the Mellon Center at the Mellon MS Center at the Cleveland Clinic Foundation. In addition, he's a professor of medicine and I've got this name, the Hazel Pryor Hostetler Chair of Neurology at the Cleveland Clinic Foundation. He is also the chair of the Society's Research Programs Advisory Committee, which is the senior group of scientific and medical advisors who help guide the Society's research strategy. In addition to all of that, Rick has a distinguished record in MS research, having been one of the pivotal leaders of clinical trials for Avonex and Tysabri, among today several of the drugs that he's been involved with. Next to Rick is Dr. Jugnu Jain, who joins us from Vertex Pharmaceuticals, a local biotech here. Jugnu is the MS disease area expert at Vertex, which is a great title because what it really involves is building collaboration, being an internal champion for MS. I forget when we first met, Jugnu, but it was a while ago.
2006.
It was 2006. I've forgotten already. Wow.
He's unforgettable.
I'll slip you 20 later. In addition to sort of being an internal champion, Jugnu is also pivotal in building collaborations with outside groups, such as the MS Society, Society to Cure, and a number of other groups. Next to Jugnu is Dr. Doug Brown, who comes to us from the United Kingdom. He is the head of the biomedical research programs for the UK MS Society. He also leads their MS Clinical Trials Network, which is a great example of a collaborative effort amongst academic researchers. And he also leads their intellectual property efforts, making sure that all of the investments that the UK MS Society makes actually reach some potential. So thank you for joining us, Doug. And lastly, we have Dr. Danny Reich, who leads the translational, I had to write this down, translational neuroradiology unit at the National Institute of Health, at the National Institute of Neurological Diseases and Stroke. Say that 5 times quickly. Danny has an MD PhD he received his PhD from Rockefeller and his MD from Cornell. He assures me that the more important of the 2 degrees is the PhD, he says. So I will see. Interestingly enough, there are 3 alumni of that MD PhD program in this room. So we have Danny, Phil Yeager, and also Brian Wong, where he is from my side. So we have it's not often you see that many MD PhDs in the room who are alumni from the same program. Danny is a specialist in both neurology and radiology, and he specializes, after some training with Peter Calabrese, one of our Promise 2010 MS researchers at Johns Hopkins, in applying MRI to clinical trials and drug development. So welcome, Danny. So thank you. So Rick, perhaps you could share from your perspective some of your thoughts about the highlights about, where are we today with progressive MS, and what might the future potentially look like as we think about research and trying to find new treatments for people with progressive forms of MS?
I think we're at the early stage of developing effective treatments for progressive MS. So we talked today about the need for having a sensitive design to test new therapies that will protect the brain and slow down progressive MS, maybe stop it, or maybe even prevent it. We talked about the animal models that might be useful in testing at the preclinical level. We talked about some of the things that we don't know. I think it's very important for people here to realize that there are a lot of things we don't know yet. We'll find out, but we don't quite know. One of them is that the gray matter, it turns out, which is the outer part of the brain, is much more effective than we ever realized because we don't see that on the MRI. We just see the inside piece, the white matter part. So we talked a lot about what's going on in the gray matter. It turns out that the problem in the gray matter is what's causing the disability. We didn't know that 5 years ago. And now that we know that, we're all focused on how we measure that so that we can treat it. So I would say that where we are right now is we have to try to understand 2 things. And we talked about this today. One is we have to understand why MS differs so much from one person to another. Why does somebody have a progressive MS case, but they really are only mildly affected, don't change much from year to year, and another person gets worse every few months, they're worse and worse and worse. We need to understand that because that'll teach us what's driving this progression. And we have to understand how to treat it the way we learned in 1980, how to test drugs for relapsing MS. So I would say to wrap this up, we're at the 50-yard line. We're going to get into the end zone with MS. We are going to cure it, but there's still a lot we don't know. And so I think that people need to keep the faith and remain somewhat patient while we sort through this.
So Jugnu, from your perspective as someone who thinks about drug hunting and trying to fill a gap, what was your takeaway as we think about progressive MS in terms of how organizations like yours and some of the others in this room might be looking at progressive MS and bringing those treatments faster to people who have it?
So I think this meeting is a very long overdue meeting in my mind because I came into MS rather late. So you have the perspective of 25 years. I got into this area only about 5 years ago. So I started impatient. It seemed like everybody knew everything about MS. T cells do this, B cells do that. So how is it that we haven't been able to stop this disease? So right from the beginning, we decided that we would not focus most of our effort on relapsing remitting disease because it seemed like many good companies and many good people had already gotten into that. So we were into progressive. And as Rick said, we know so little about the disease. We don't have good targets. A drug company needs targets. What do you treat? We don't have very good models, animal models. That I think is only part of the problem because you can still create models around the target that you're going to work with. But the biggest hassle is how do you run clinical trials? Which patients do you treat with what mechanisms? What readouts do you use? Because you can't go into people's brain and take out a piece of that. You can't have blood markers that reflect the brain disease unless you have verified them ahead of time. The MRI imaging that is used very successfully in relapsing remitting MS is likely not to be that helpful in progressive MS patients because it's very non-specific. It just tells you there is a lesion. It doesn't tell you whether this lesion is because of edema, whether it is because of inflammatory cells, whether you lost neurons, whether you have new myelin. It's just a spot. And you have to wait for months and months to figure out whether the spot became a black hole, as it's called, or became remyelin, or whatever happened, disappeared. As we get into progressive MS trials, I think one of the things the society can do, particularly, is not just stop at a meeting like this, but really build consortiums that are cross-industry, cross-academia, cross-FDA, build the group that will tackle this disease together because we can't afford to do it independently, one at a time. And I think for the guidelines that need to come on how to do clinical trials, FDA will need to be involved and buy into it. And that is what will give companies like us some advantage in reducing the risk because MS trials are long enough for relapsing remitting where we know what to do. In progressives, we don't even know how to design the trials. So I think that's the session for tomorrow. And I know we will get into a lot of vigorous debate. And I think that debate needs to be carried through to a point where we have some action items associated with it.
No, absolutely. I absolutely agree with what Jugnu was just saying there about a need for a coordinated effort, and I think it needs to be a coordinated international effort. I think one of the challenges that everyone will face, including the UK MS Clinical Trials Network at this point in time, is to decide how best to design clinical trials for MS and how best to measure the potential effect of neuroprotective therapies. We just don't know how to do that at the moment. And I think the good news is that a lot of research has been done to get us where we are now, but we just need to bring that all together on an international level to get that data in place so that we can move forward with clinical trials of neuroprotective and potentially repair therapies in MS. We've learned a number of things through the UK Clinical Trials Network. We don't have the biomarkers we need, but we've spoken about it in the past, and we spoke about it briefly today, that the samples are out there from people, so we just need to bring all those samples together. We need to correlate that with MRI measures, other measures as well, so that we can really identify the biomarkers in the blood, in the cerebral spinal fluid, that will really make a difference in helping us measure drugs that will work. We also need to come up with better clinical trial designs, and trial designs that will speed up the identification of potential therapies and do it in a more cost-effective way. Now, these trials are extremely expensive. We could potentially start by learning from other conditions first. So there are many drugs, many hundreds of millions of dollars spent on trials in the stroke field, for example. And they're drugs that look to try and find a neuroprotective effect, but in a very acute, massive, damaged condition. Whereas those drugs that have failed in stroke may actually be beneficial for MS. So we could learn from that and translate it into MS. A lot of those drugs could be off patent, a lot of those drugs could be fairly cheap. We could actually plug them into a system and compare drugs together to try and identify drugs in a pretty short period of time. Now, there are no guarantees, of course, but I think we could be working together, and I hope that we can work with the American MS Society and other MS societies as well to drive this forward after this meeting's finished tomorrow.
So continuing the theme of collaboration, Danny, you did your training with Dr. Peter Calabrese at Johns Hopkins, who was one of our Promise 2010 team leaders. It was a large collaborative group, and your focus was on imaging and how you can find new ways to probe into the brain to see how repair is happening, how neuroprotection is happening. So put on your hat as a think speculatively for us. What kind of tools do you think we might have in the next 3 to 5 years or even 10 years that could allow us to look and perhaps begin to understand the brain more specifically as it relates to progressive MS?
That's a great question. The tools we have are good. I disagree a little bit with what you said about how MRI is not a great tool for looking at progressive MS. I think Rick and others have shown that it can be very, very useful. But I agree completely that we don't know what to look for in there. I think there are new tools coming down the blocks, new ways of setting up our MRI machines to probe different problems in the brain and spinal cord, and new imaging techniques, molecular imaging techniques that will really be incredibly specific for picking up various aspects of pathology. But I think perhaps more than anything else, what we can do over the next even 2 to 3 years is get the data with the tools that we do have now. And from collection of data on the same people who, as their disease continues over the next 2 or 3 years, we can learn a tremendous amount, not only about how the disease is different across the population, but how its rate of change over time is different. And I think that's going to be the thing that is going to let us figure out how to do these trials. And I would say one more thing, just in follow-up to what I heard before, is that I think it's obviously critical that we need to understand the disease better in order to focus our trials and not do wasted trials. But it's really a two-way street. And if you think about clinical trials, and the way we at the NIH like to think about it and have had success thinking about it in the past, is that they are essentially the equivalent in human beings of an experiment. And so if we think about clinical trials as a scientific experiment that's well controlled, that's carefully done, where we can learn not only does the drug work, which is of course the most important question, but from the results of the trial, how the disease is happening and how the disease is causing disability. From each trial, even the ones that failed, we may learn something. And I would just put out a plea to all of my colleagues in the research field that as we design these trials, we need to have a plan up front for how we're going to be able to use the data, how we can collect the data, and how we can build into these trials some way of learning, even if the trial fails for whether the drug works, something about the disease that we can use the next time around.
you. Before we open for questions, I do want to ask Rick for perhaps if you could share some perspective, because you opened your remarks about starting in the MS community, not having any therapeutic options. You were there when we went from having some potential therapies to now having several of the ones that are now, everything that's approved now. And so in your lifetime, you've seen the field shift. And so perhaps if you could give us some perspective as a clinician as to how you, when you see patients, what's different about your conversation with a patient today? And, what would you hope that conversation will look like, say 5 years from now, where our strategic response, has been implemented and we perhaps realize some of the fruits of this conference?
Well, it's an interesting question. For many patients, we have shifted from saying, we're not sure what this is, we hope it's nothing serious, we'll monitor this, because we had no treatments. We've now shifted to, we know what this is treatable, we're going to take care of you and you'll be okay. So a lot of my practice now has shifted 360 degrees or 180 or whatever it is, from this denial and deferral and optimistic hope to proactive action. And so now it's nice to make a diagnosis early. It's very hard on the patient, and there's an adjustment period, but we can do something about it. Totally different than when I started. On the other hand, we have a horrible unmet need, which is another group of patients for whom we can't say that. We can say there are many things we can do. We can help you adapt, we can treat various symptoms, but we have no way of really stopping this. And that's still hard. What I tell those patients is that we're going to stop it. I just don't know when. It's a matter of research. So it's kind of a bittersweet sort of thing. We have made so much progress, but it doesn't apply to everybody. Now, one thing I would comment on briefly is, why is this so hard? Why should this be so hard? If we got to the 50-yard line, why don't you just do what you did to get there and get into the end zone? Why is it difficult? It turns out that MS is more than one thing. It's an inflammation problem in its beginning, and we have good treatments for that. But in its midlife and late life, it's not an inflammation problem anymore. At least it doesn't seem to be, and it certainly doesn't respond to inflammation treatments. The problem is that the nerve cells wear out, they die. It's called neurodegeneration. It may be similar to neurodegeneration that we see in other conditions for which we don't have a treatment, such as Parkinson's or Alzheimer's or ALS. We don't know that yet. This is one of the most difficult problems that we face in medicine, neurodegeneration. So it's a whole new problem. The answer isn't that we haven't been trying, it's that it's a much bigger challenge. Thank you.
The question is how do we get companies that are doing clinical trials to essentially share their data, as you suggested they do?
Yeah, I'm not a company man either. But
He says he does work in Washington.
I do work for the company. It's a great question, and I was hoping somebody would ask that. I think a major part of that has to be pressure from the community of patients and loved ones who care about the disease to make these requests, demands. And I think in the end, it's really a win-win, and I think we have to understand it as a win-win from not only from the researcher's point of view, but from the drug company's point of view. I think the more that can be learned about what we call the natural history of the disease and what underlies the differences that we see, the easier it will be to find the targets that are necessary. And I think if it's sort of understood in that way, then this could be accomplished. Um,
So the question is, who do we pressure? So,
Right.
who do we pressure, Andy?
Well,
Look to your
you know,
right.
no, and I think the NIH can the NIH can play a big role in this, but the MS Society may even be able to play a bigger role in, because of the connections that have been made with all the various stakeholders. That, you know, this, and I'm now speaking in very general terms, because I don't have all the answers, but I think there are examples of conditions, and I think Alzheimer's may be a good example of this, where drug companies have come together at the table and made investments, shared investments, in understanding that disease, that are now beginning to bear fruit through initiatives related to imaging, related to biomarkers, and they're beginning to bear fruit. And I think we could do something similar to that in MS, and I think the MS Society and Fast Forward could play a major role in that.
So the question is are there drugs that can help speed up the rebuilding of axons and nerves in MS? So Rick or Danny, would you, or the panel?
Over to you.
I think the short answer is no. We don't yet have a drug that is known to promote repair in the central nervous system. That is the goal. We don't yet have a drug that is known to slow the process of neurodegeneration. That's also a major goal. I think you're on to something really important. I think one of the early pathologic changes in MS brain is that the synaptic density, which is the number of contacts between one nerve cell and another, begins to go down. And we don't know why. But if we could find a way to increase synaptic density, I think that would be a major therapeutic breakthrough. So you are onto a really important question. We don't have an answer to it right now.
What is that?
There is a treatment which will promote remyelination. So while we do not have the ability to regrow dead nerves, if you can take nerves that got demyelinated, that lost the insulation that would have helped them conduct electricity, if you were to rewrap them by new oligodendrocytes that do that job, you should keep them, and you should keep them, you know, functional. And there is the new treatment that is just started in clinical trials, and if that works, then it will delay disease progression or will block it at that stage for whichever neuron or whichever axon hadn't died yet.
Just to add to that, there's a theory in MS that the problem in the progressive stage of the disease is that the brain doesn't naturally repair properly, meaning that the remyelination doesn't occur. And the reason people recover in the early stage of MS but not later on is that remyelination is more effective in earlier stage disease. So I think that the approach that we're hearing about is to use a pharmaceutical to promote the remyelination in people with later stage disease. If that works, that'll be a huge breakthrough. And I think the point is that's in the testing phase. So we're all keeping our fingers crossed because that could be a big breakthrough.
I think one observation on this also that is different, you know, now is that the ability to have these kinds of large-scale studies, the large-scale kind of analysis of data that Danny was referring to and that Rick referred to, is also really an emergent theme of the last, let's say, 5 to 10 years, and that we now have the tools in which, if you have mountains of data, you can analyze that data, which we couldn't do probably 15 years ago. And so the ability to do this kind of massive genetic analysis will undoubtedly get us past that 50 yards a little faster.